just say that although Dr. Mushatt hardly needs an introduction, let me mention that he is a professor of medicine and chief of the section of infectious diseases. He had received his medical degree in 1986 from Harvard Medical School, completed his internal medicine residency training at the Harvard-affiliated Brigham and Women's Hospital in Boston, and then completed his ID fellowship at Tulane. During the second year of his ID fellowship, he also obtained his master's in tropical medicine and at the School of Public Health. Dr. Mushatt is well known to us. He's been a leader in infectious disease and also PI on multiple studies, including the International Network for Strategic Initiatives in Global HIV Trials and NIH-funded research group. Dr. Mushatt loves teaching, and he's appreciated as a wonderful teacher and a wonderful colleague and consultant. And without further ado, I will turn it over to him. I want to remind everyone, please, if you have questions, type your questions in the Q&A box, and we'll hope to have a discussion after the presentation. Thank you, David.
Thank you so much, Dr. Bateman. It's a pleasure to be here. This is my first grand rounds on COVID since January. And wow, has there been a lot of new information. In fact, this morning, every day, there's something new. So forgive me, it's a lot of slides, but there's just so much to tell you about. I'll be very happy to share these slides. You can either email me and I'll give them to the Department of Medicine. So today I'm going to cover epidemiology, current treatments, potential new treatments, vaccines with a focus on the Delta variant and the third dose / booster, a little bit more about variants and what we think may be the end game here. So this is the Johns Hopkins website from 2 days ago. It shows the number of cases around the world. You can see that there have been over 200 million cases, and some experts say there may have been 10 times that many. Many go undiagnosed, or perhaps as many as 2 billion or more. Sadly, more than 4 million people have died, probably more than that, and nearly 5 billion vaccine doses have been administered. In the last 28 days, there have been about 18 million cases and over 250,000 deaths. If you look at the US here, you see we lead the world in total numbers at about 37 or 38 million, but again, we've probably had many fold times that have, many of which have gone undiagnosed. Followed by India at 32 million, and then down here, Brazil at about 21 million. Iran is here because of the large number of cases in the last 28 days. So let's look at the US. This is my favorite website, the New York Times COVID site, great site, really good graphics and information. And what you can see is that the US is in the middle of this fourth surge. It looks like it might be starting to taper a little bit, but certainly the number of cases are up over the last 14 days, tests, hospitalizations, and deaths. The good news is that the vaccination levels are improving. And if you look at people over 65, it's over 81% who've been completely vaccinated. And for all ages, it's about 52%. And for adults over 18, it's 63%. The problem, of course, is that while the cases may start to stabilize, the deaths are going to keep going up for a while. Here are the hotspots in the US. This is defined as average daily cases per 100,000 people. And purple is the most intense number of cases. You can see it's confined largely to the Southeast and the Northwest. There's relative sparing of the upper Midwest and the Northeast, but you are seeing encroachment on these areas. This is the data for Louisiana, and you can see that the good news is that in terms of reported daily cases, it looks like we plateaued or going down a little bit. I'll believe it when that's sustained. And again, while the number of cases may be stabilizing and go down, the hospitalizations are still high and the death rate is still going up because deaths tend to lag a couple of weeks from onset of diagnosis. If you look at the vaccination rate in the state, we are doing better than we were a month or two ago. For all ages, we're at 40% fully vaccinated. For those 18 and older, 50%. And we're doing quite well with the 65 and older. Nearly 90% have received one dose, and almost 80% are fully vaccinated. Here is the COVID vaccination data for the state of Louisiana by healthcare region. We're in region 1. New Orleans Parish shares that with St. Bernard, Jefferson, and a few other parishes. So we are doing the best at about 52%. Not too far behind is the Baton Rouge area at 43%, but you can see that southwestern Louisiana is still below 30%, and the northern regions are still in the low 30s. So we have a long way to go in Louisiana. So if you look at the number of doses being administered per day, this is also from the New York Times, you can see that the good news is that for the US as a whole, it is picking up a little bit. We're up to nearly 900,000 doses per day, but it's a far cry from the 3.5 million doses that were being given daily back in April. Here's some good news for Louisiana. This is actually 2 weeks old. It hasn't been updated, but 2 weeks ago, if you look at the daily number of vaccine doses administered per 100 people, we led the country. We were ahead of everybody else. Now, of course, that was worked out to roughly a threefold increase in our vaccination rate, but our vaccination rate had gotten very low. And so 3 times a low number is still a low number, but at least the improvement is there, the trends are there, so everybody's hard work is paying dividends. Here's the dashboard for New Orleans. You can say we're still having several hundred cases a day over a 7-day average. The positive test rate is still high, though it's lower than it had been. We've got about 2,900 people in Louisiana hospitalized. And you can see again that Orleans Parish is doing better than most of the rest of the state in terms of vaccination. We're at about 55% who have completed vaccination out of the total population and nearly 70% of the adult population. So we're making good strides. If you look at the number of new cases, the 7-day average is going down. This is heartening. If you look at for Louisiana as a whole, this is the data for hospitalizations, and you can see that we have plateaued over the last week or two. A little bit of a decline, another plateau, but this is, certainly still an awful lot of people in the hospital. It's not going down quickly, and so this gives pause for concern, and I think this is reflected in our local hospitals. Here's a data for hospitalization and ventilator use just for New Orleans, region 1 actually, so that also includes Jefferson and several other parishes. You can see that the green line is the number of COVID hospitalized, which is peaking and plateauing, and it looks like the number of people on ventilators is plateauing as well. Here's the Tulane University dashboard, which is updated several times a week, and you can see that the numbers are starting to creep up. 2 days ago on Monday, there were 10 new individuals who tested positive. We've been creeping up for a few weeks. At 1% for that day of the total positives or the total number of tests, we're still well below the New Orleans and Louisiana averages, but we do have 61 active cases. And with 2,000 new freshmen, I think it may be a record number or one of the highest numbers we've ever had of new freshmen who just arrived in this last week. There's certainly probably going to be some importation of cases and some increases. And so we hope we're prepared for this. So here's the ensemble model that the CDC uses. This incorporates multiple other models from the US and other places. Looking at forecasting COVID cases, deaths, et cetera. And so I'm focusing here on cases, weekly cases. And you can see that for the US, it looks like it's going to be a while before the US peaks, probably continue to go up through September. The good news is that at least in Orleans County, or parish as we call it here, the forecast for the weekly cases is a downward trend, a fairly steep downward trend. Hopefully this will hold up. We're at about here. This data is already a little bit old and gets updated every week. So hopefully this will hold up. So let's turn then to what's probably most on people's minds, perhaps second only to vaccines, and that is current treatment. What are the currently available treatments and what do we recommend? So first of all, the CDC has this thing called the Coronavirus Treatment Acceleration Program, which has 630 plus drug programs in planning stages or in development, over 460 drug trials that have been reviewed by the FDA. There are 11 treatments that are currently authorized for emergency use, the so-called EUA, and there's one that's approved by the FDA for use. And you can see in terms of the types of treatments being studied, there's quite a range. There's antivirals, there's cell and gene therapies, immunomodulators, neutralizing antibodies, and other approaches. For instance, we had a trial here that was part of a larger group that was looking at an oral protease inhibitor called Camostat, which unfortunately did not pass muster. And then there's combinations of regimens. So there's a lot of drug development in the US and elsewhere. This I like this graph. It really kind of summarizes perhaps an oversimplification, but it gives you a pretty good sense of what this disease is all about in terms of the pathophysiology. So COVID-19 is a disease of viral activity early on, and then the host immune response. So early on, probably several days, 2 to 3 days before an individual actually has symptoms, they can have detectable viral load or virus. It peaks in the first few days, it wanes by about a week. And during this later period, the antibodies are starting to go up. And this is when we start to see some of the dreaded complications, such as ARDS, ICU admission, respiratory failure, clotting, et cetera, which is probably more due to the excessive or hyper inflammatory response, both in terms of the antibodies, cytokines, and various other immune mediators. It's a very complicated paradigm, and I still, after a year and a half, don't think anybody really knows for sure exactly what's going on, but this is a pretty good model. So what we glean from this is that antivirals are going to work best probably when they're given early, before the viral load gets really high or before it falls off and the immune response kicks in. And then immunomodulators will work either somewhat early to prevent the hyperinflammation and or once the hyperinflammation ensues. So here's a wonderful algorithm which was crafted by our fantastic PharmD Wes Pierce in the TMC pharmacy. I love this. Feel free to share this. He got some input from some of us in ID, and of course Josh Denson and others in pulmonary and others. We had this last year, but it's been updated recently. So basically, what this shows you is when to use the different treatments depending on the oxygenation status of the patient. There's really, for patients who have O2 sats above 94%, there's no antivirals or immune modulators, but this is a good time to use monoclonal antibodies to prevent progression. And we'll get to that in a minute. So the main intervention here is going to be steroids. Steroids have been proven pretty well to reduce the risk of dying from this disease, but they should not be given to people that have relatively good oxygenation. They should be on oxygen or have oxygen saturations of 94% or less or worse. Remdesivir, of course, is FDA approved. It is generally used for the milder, earlier stages, those who have severe COVID-19 but are not on high flow oxygen. They're either on oxygen, have oxygen saturations of less than 94% or so, or relatively low flow nasal cannula. There is debate about this. The WHO does not recommend it. There are hospitals in the area that are not using it. It is sort of a marginal benefit, I think. Now, the newer interventions are the immunomodulators. There's baricitinib and tocilizumab. These are both now recommended by the NIH and the ID Society of America in their guidelines. I will say the indications are a bit murky, and don't feel badly. I actually queried two of the co-chairs of the NIH guidelines committee, Drs. Henry Mazur at the NIH and Clifford Lane at the NIH. And to be honest with you, they really didn't have any pearls. They said, data is very limited. It's hard to know, which one to use. Is one better than the other? They've never been compared head to head, so we don't know that. But the bottom line is these drugs seem to be beneficial if they're used relatively early, that is within 3 days of admission or within a day of ICU admission, and generally speaking work better in those that have elevated inflammatory markers. Now, if there's no consensus on how to define that, but for tocilizumab, one of the studies did use a cutoff of CRP of at least 7.5 milligrams per deciliter as their cutoff for what they considered to have to be high elevated inflammation. The key here is using them relatively early, particularly in patients that are progressing very quickly. Not your stable patient with low inflammatory markers, but the ones that come in and they progress very fast, they probably will benefit from these. Unfortunately, tocilizumab has a huge national drug shortage. We have very little here at Tulane. I know at Rapides and Alexandria, they have none left. Baricitinib is hard to get. It's just not an adequate supply. And so this is creating a lot of angst for us. So let's take a look at the individual components here. So remdesivir, I don't want to beat a horse into the ground. It's been around a long time, but I think most people know that the data so far shows that it seems to reduce the median time to recovery by about 5 days. And in a subgroup analysis in this ACT-1 study, there did appear to be a mortality benefit, 4% versus about 13%. But it was a subgroup analysis. It has not been replicated in other studies. So I'm a little skeptical that there's a mortality benefit. I think it's possible, but I'm not convinced. So therefore, some people are not very excited about remdesivir. Dexamethasone, I think, is pretty firmly entrenched. This study looked at 28-day mortality, and it was published in the New England Journal. This is a forest plot, and what you see here is that for all patients, there was there was a rate ratio of 0.83, which is about a 17% reduction in 28-day mortality. It was even better, about 36% reduction in mortality for those on invasive mechanical ventilation, and not quite as good for people on oxygen, but it did not help those who are not on oxygen, and it may even cause harm. So if you use it too early, it might actually blunt the immune response, prolong viral persistence. But for those who are sick on oxygen, et cetera, it does seem to be beneficial. So what's newer, of course, are these immunomodulators. So the first one, tocilizumab, is a monoclonal anti-IL-6 receptor antibody. IL-6 levels, we used to measure those last year in some of our patients when we could get them. They tend to be high, but they're really not very helpful in management. But the idea here is that this drug, which has been used for autoimmune diseases, can block these, reduce the effect of these monoclonals. And in this study published in the Lancet, they looked at 28-day mortality here on the left, and you can see that going out to 28 days, the tocilizumab did have a statistically significantly lower, about a 15% lower mortality in this population. And then if you look over here at the 28-day mortality by baseline group, what you see here is that in those who were on steroids, there was a mortality benefit, but if they weren't on steroids or had not received them, they did not. So the recommendation is to generally add this to steroids. Some people actually start steroids to see if they fail steroids, then they add it. Okay, baricitinib is the other immunomodulator that's in use. It's a Janus kinase or JAK inhibitor. It reduces cytokines that have been found to be elevated in this disease, interleukins, interferon gamma, GM-CSF, et cetera. This is a study, it's a retrospective study, so it's not the greatest, but it does gives us some insight. If you look at the Kaplan-Meier survival curve for 30-day mortality, you can see the blue line here represents baricitinib plus dexamethasone versus dexamethasone monotherapy. There's a pretty significant increase in survival probability. And then if you look at this study, which has been posted to the preprint server MedRix IV. Now, of course, this has not been peer reviewed yet, so take it with a grain of salt, but it is a double-blind randomized study. And here on the left, you have two populations. Population one is all participants, and this population down here too is patients on oxygen supplementation who were not on steroids. And the baricitinib group, you can see in red, does have a significantly lower mortality out to 28 days in both groups. If you look at the forest plot here, you can see that in the overall population one, there's about a 0.5 hazard ratio, so about a 50% or 43% reduction if you look at this population, and for this population, it's closer to 70%. So it does seem to help. I think most people would say that you can use it in lieu of steroids if for some reason there's a contraindication to steroids, so you can't get them, but I would probably add this to dexamethasone. Monoclonal antibodies are quite hot right now. They're exciting and they're really quite effective, and we need to be using them. They're definitely underused. First of all, if you want to know where to get them, there's two locators. There's the Health and Human Services locator, and there's this one from a national infusion company or center that has it. They're pretty good. They're not always verified, but they give you a pretty good sense. So for instance, in New Orleans, you can get them at Tulane, Oxnard, I think at East or West Jeff, University Medical Center. There's some, and there's some south of here in the Bayou. There's one or two in the North Shore. There's a number in Baton Rouge. You're going to have to call, but to confirm that, but they are out there. And in addition, now that people are starting to give them subcutaneously, they're going to be more widely available. I believe the FQHCs, federally qualified health centers in this part of the state, are beginning to deliver those or deploy them. So what are the indications? So for the most part, these are to be used in non-hospitalized patients who are not very sick. They should not be on oxygen. They shouldn't be desaturating. They should be relatively stable. However, there is a carve out for hospitalized patients who come in with something other than COVID-19. So we've had people who came in for a broken hip. They're elderly and frail, they're high risk, and they happen to test positive they can get it, because the key here is getting people early to prevent progression. So bottom line is they need to have mild to moderate, not severe. They should ideally not be on oxygen, although they can be on oxygen if they're on home oxygen, as long as there's no increase in the flow rate. They should have lab confirmed infection, and they should be at high risk for progression to severe disease based on the following criteria. These are from the NIH. So the top category are the really hard and fast ones. These are best represented in clinical trials, and these are softer, but they're still widely recognized. So basically, anybody 65 and older, anybody who's has a BMI of over 25, so that's a lot of people. It's probably more than half the people in Louisiana. Anybody who's overweight, anybody with diabetes, heart disease, chronic lung disease, chronic kidney disease, pregnancy, sickle cell, and many other conditions those are all associated with the higher risk of progression and death, and they are eligible. Another very important clause here that that's often overlooked is the FDA and the EUA also recognize that there may be other factors such as race or ethnicity that place individuals at high risk. So many of us interpret that to mean probably all minorities, all African Americans, perhaps all Hispanics, certainly all Native Americans. They have very, very high risk of progression. And I think most people are recognizing this. It's a little bit of, the language is a little wishy-washy. I think the FDA did that intentionally to give you some wiggle room depending on supply, but we need to, in terms of health equity and fairness, we need to make sure that everybody who's eligible are getting opportunities to get these. What's the data? The data is quite good. So this is for treatment, not a prophylaxis. And here's a study published not too long ago in Clinical Infectious Disease, mostly got the older bamlanivimab, but some, about 20%, got the Regeneron product. And you can see there was about a 50% reduction in hospitalization. That's tremendous. I don't know of anything else that works that well. And then there's this press release. And obviously, we don't want to be practicing medicine based on just press releases, but it's the only data I could find from this trial, this Regeneron trial in non-hospitalized patients. It showed about a 70% risk reduction, regardless of the dose. There were several different doses that were being looked at. So these monoclonal antibodies really work. We've all anecdotally seen people get these, and they often feel better by the next day. I've seen it. I believe it. They work. And then the newest indication is now monoclonal antibodies for post-exposure prophylaxis. And this is for people who, again, are at high risk, similar definition as I just mentioned, and who have the following vaccine history and exposure history. So they either are not vaccinated or not fully vaccinated, or they could be fully vaccinated, but if they're, for example, a kidney transplant patient, we would predict that they would not respond very well to the vaccine, and therefore they should get monoclonal antibodies. So any reason, such as immunosuppression that could impair significantly the vaccine response is a reason to give them this if they've been exposed, and they have to have an exposure history to SARS. So again, I suspect this is being underused. This is a recent article from August 4th in the New England Journal. This is a nice study looking at the sub-Q Regeneron cocktail to prevent COVID-19. This is a post-exposure prophylaxis. And here you can see the Regeneron versus the placebo line. There was about an 81% relative risk reduction for symptomatic infection. And symptomatic infection is a tough barrier because most, for instance, vaccines tend to work better against death, hospitalization, than they do against infection or symptomatic infection. You can see on the right here, especially on this left column, the Regeneron combination had significantly better or shorter duration of symptoms, whether asymptomatic or symptomatic, and duration of high viral load. So this really works. What are some potential newer treatments? There's a number of things out there. A very basic one here is inhaled budesonide, which of course is readily available. It's used for people with asthma and COPD, and there's been some signals over the last year that it might be helping. This is a randomized controlled but open label study that did show what appeared to be better, faster recovery in individuals who received inhaled budesonide versus usual care. Note that this is open label, so there is potential for bias here, but it would be very nice if we had something simple like that we could give our patients and just call in for them if they call and say they're positive, and something to kind of nip it in the bud. Another thing that's new that may be approved or at least allowed in the coming months is the use of the monoclonal antibodies for hospitalized patients. And in one of the studies, what they found was that the antibody cocktail did not work in general for people who were hospitalized with severe COVID, with the exception of a subgroup who tested negative for anti-SARS-CoV-2 antibodies. It seems that a small percentage of people who get admitted don't have the antibodies upon admission. Perhaps they develop them later, or perhaps they have a defective immune response. And they did seem to benefit, and that kind of makes sense. If they're not responding well, it would make sense that giving them supplemental antibodies would be helpful. And this may be coming out at some point as an EUA. My understanding is that the dose is much higher. It's about 4 grams, so it will be a lot more expensive and will use up a lot more of the drug. What we really need, though, to turn this into a more of a flu-like illness is an oral drug that we could call into our local pharmacy and give to people, just as we do with Tamiflu. If I have somebody who calls me during flu season and they say, I think I have the flu, and if they tell me they have fever and chills, I don't even bring them in. I don't want to expose other people. I don't want them to get, to have to spend the money on seeing me and getting tested. I just call in Tamiflu. It's easy. Well, this is a drug that may get us there. Molnupiravir is it's actually, I believe, was invented by Emory, and it's now, I don't know if it's licensed to, but it's being handled by Merck. It's an oral ribonucleoside analog. So it basically inhibits replication of the virus. And in this phase 2 trial, which is again on the preprint server, it's not been peer reviewed yet. It did show faster viral clearance based on nasal swabs. Now that, may or may not translate into better outcomes. That remains to be seen. But the hope is that this may be useful, particularly very early. Again, I could see this as being something that one could use early, that one could get for a patient rather than having to bring them in. Now, ivermectin, of course, is kind of a thorn in our side. This has been plaguing us for the last year and a half. It's been used off and on. People were using a lot of it last year, and it's sort of coming back into fashion. There are many of these, what I would call rogue groups and websites that tout its benefits. And so, there's many studies, not very well done. Some have shown a mortality benefit, but look at this. On July 14th, one of the key articles that other studies were hinging on was withdrawn from the preprint server because of allegations of data fabrication, plagiarism, and ethical breaches. Oh my goodness, not good. So, this gives us real pause. And this is one of the better studies. This is a study published in CID not too long ago. It is a systematic review and meta-analysis, so it has all the caveats. But basically, they concluded that ivermectin did not reduce all-cause mortality, did not shorten length of stay, or improve viral clearance, at least in the randomized controlled trials in patients with mostly mild COVID-19. So I would conclude that this drug is probably not going to be effective. There is a large study being conducted in Brazil. I think they're trying to enroll over 30,000 patients. And so studies like that may give us better answers. It's probably a relatively safe drug used at proper doses. So if physicians can use it off label, because it is FDA approved for parasitic infections, and therefore an MD can prescribe it. But I would be careful. And you probably have seen recently, there have been many reports of poisoning in individuals who procured the veterinary form of ivermectin and took large doses and had seizures and other complications. So beware. What about anticoagulation? I get asked this a lot, and I always tell people I'm an ID specialist. I'm not a hematologist. I'm not a hospitalist. I'm not a thrombosis expert. But, I thought I would take a brief look at it. So we all know that thromboembolism is more common in COVID-19. In this particular study in eClinical Medicine, the overall venous thromboembolism rate was about 20%. It was higher in the ICU. DVT, 20%. PE rate was about 13% higher in the ICU. Fortunately, arterial thrombosis seems to be relatively rare. So this is something we are all aware of. It's theorized that some of the microemboli, microthrombi may be contributing to the ARDS and respiratory failure. What's been disappointing, though, is that there have been many studies looking at anticoagulation, and very few of them have shown any benefit. So here the question is therapeutic anticoagulation better than prophylactic? Maybe. So in the August 4th edition of the New England Journal, there are 2 studies that look at therapeutic anticoagulation with heparin in non-critically ill and in critically ill patients. Now, in these groups, about 90% of the subjects received low molecular weight, and the other 10%, I believe, received unfractionated heparin. And what you see here is that if you look at the overall group, these had moderate disease, the adjusted odds ratio statistically significantly favored the intervention in terms of organ support free days. So the probability of superiority of therapeutic dose anticoagulation was about 99%. So that would suggest that in this population, it does work. If you look at the accompanying article or study, these people also received primarily low molecular weight heparin. You do not see a benefit. The adjusted odds ratio, the confidence interval crosses 1. There's about 100% almost probability of futility or a 95% probability of inferiority. Same thing for survival to hospital discharge. There's an excellent editorial in that issue of the New England Journal by a scientist from the Netherlands, who I think it comes out being very lukewarm about therapeutic anticoagulation. And he basically says you need to balance the risk of bleeding. You can see here in this particular study, there was a 3.8% incidence of major bleeding in those who received therapeutic anticoagulation versus 2.3% in usual care. That sounds small, but that's people that bled and may have died. So he's very lukewarm about it, and I think more data is needed, and I will certainly defer to our hematologists and thrombosis experts. I will say that in I think the most recent New England Journal, there's a very interesting review of heparin resistance. I was not familiar with this, but there is this phenomenon whereby heparin has been shown to there's sometimes resistance to the effect of heparin in some individuals where you can't seem to anticoagulate them. And the authors, opine that this may be part of the problem in COVID, where the cytokine disarray and the milieu in these patients may be such that the heparin is not working as well. And so better interventions clearly are needed. All right, vaccines, foremost on people's mind. So these are the leading vaccines around the world. We have 4 of them, or 3 of them, and one more in development. We have the Pfizer and the Moderna, both mRNA, the J&J, which is adenoviral, and then the Novavax is another US vaccine that's been in development, but unfortunately it still hasn't received authorization or EUA yet. And my understanding is that while it has shown over 90% efficacy, they are having some significant problems with manufacturing and production. And they are so far have not been able to satisfy FDA requirements for manufacturing quality and standards. And I don't quite understand the technical aspects, but it sounds like this is a potential deal breaker. But we're hopeful that they will, because we can use more vaccines, particularly if we can send some overseas. So why are vaccine efficacy numbers so variable? We're all going out of our minds. I get so many emails about this, how effective are they really? I see different numbers. Well, there's a couple of reasons, and there's probably more. But the way I look at it is these studies vary depending on the prevalence of the mutants. So a study that's been done more recently where there's more delta in the population is probably not going to perform as well. There does appear to be waning of antibodies in those who have had COVID or in those who've been vaccinated. So it appears that there may be reduced protection over time. And then there's variable endpoints between the different studies. Some studies are looking at asymptomatic infection and or symptomatic infection, which is the same thing as disease, and or hospitalization and death. As a general rule of thumb, vaccines work best against hospitalization and death, not quite as well as against disease, and less well against asymptomatic infection. The eminent Jay Coles here at Tulane is fond of pointing out that it's really hard to block that initial infection in the nares and the respiratory tract. But once it gets there, the vaccines work better at preventing it from spreading to the ACE2 receptors in other parts of the body. In addition, these studies from around the world are done in different populations, different ages, different comorbidities, and there's also some differences in dosing intervals, which I'll show you right next. So this is a nice slide from Scobie of the CDC at the recent ACIP meeting, where they combined the data for these 4 different studies done in 4 different parts of the world. The dark blue circles are the alpha. That's the previous variant that's now been replaced essentially almost completely by the Delta variant, the red triangle. And what you see is here, it's for the most part, they have data for the infection without symptoms, symptomatic disease, and hospitalization. So in England and Scotland, in this study, you can see that the against Delta, the vaccines, the patients got either Pfizer or Moderna, 2 doses. Against the just simple infection, they didn't work quite as well, but they work much better against symptomatic disease and hospitalization. Similarly, in Canada, the difference in this case was very small between the effect on Delta and Alpha. Now, if you look at Israel and Qatar, what you see is a much bigger difference. The vaccines didn't work very well in terms of preventing infection and symptoms, but they still held up well in terms of preventing severe disease in both cases. Now, interestingly, if you look over here on the left, it turns out that the UK and the Canadians spaced the second dose of these out to 12 weeks, unlike us. We give them at 3 to 4 weeks. And experts are saying that that may actually improve the antibody response. I don't know that that's being looked at in the US for changes. It would require a lot more study, but it's an interesting little sidelight to keep in mind. But the good news here, I think, is that they work pretty well, even against the Delta virus, in terms of preventing death. Here's some data from SCOBY at the CDC presented recently to their ACIP meeting, looking at the Pfizer vaccine 6-month efficacy, overall 91%. But what you see here is that when you look further out from vaccination, when you go from less than 2 months to 4 to 6 months, you do see a fall from 96 to 84% vaccine efficacy. So this goes along with the contention that there is a waning of antibodies and/or immune protection. But overall, pretty good protection and similar data for Moderna. This is the most recent data in the US. It's from the MMWR last week. This looks at the Pfizer Moderna vaccines and preventing infection in nursing home residents, which are probably more frail. They have more comorbidities. They tend to be older, so they're a more challenging population. And what you see here is they compare 3 different periods: the pre-Delta variant, the intermediate period, and the Delta period. And what you see here is that there is a downward dose response. During the pre-Delta period, the vaccine effectiveness was about 75%, but then it drops to 53% in the Delta. So we think that that's a combination of the fact that they may not work as well against Delta, but it's also probably a time factor that, Delta is more recent and nursing home patients got vaccinated relatively early in the US, and so they may have waning immunity. So what about the vaccine 3rd dose or booster? So as you know, there was an emergency authorization issued on August 12th that allowed for the 3rd dose to be given in those who received the mRNA vaccines who have moderate or severe immunosuppression. And then the Biden administration has announced that on September 20th, all persons who are at least 8 months out from their 2nd dose will be eligible for a 3rd dose or a booster. The CDC is currently reviewing data for the 2nd dose after the J&J, but in today's news, breaking news, this is not published, it's press release kind of stuff. The sponsor is announcing that there was a 9-fold increase in anti-spike antibodies when a 2nd J&J vaccine was given at 6 months after the 1st dose. So this looks promising, but I'm sure the CDC is looking for clinical data. And then there are Delta variant boosters in trials. So the 3rd dose is currently recommended for all these people who are moderately or severely immunocompromised. So typically your organ transplant patients, hematopoietic stem cell transplant patients, patients with things like advanced AIDS, and patients getting steroids and other immunosuppressive agents. And this is something you can easily look up. Do boosters help? It looks that way. The Israeli HMO Maccabi, which is has a large database in Israel, showed 86% efficacy after a booster in those over 60 years of age. Now, I don't think there's a control group that I could find, but it hasn't been published. But the historical protection was about 55% for severe illness in those over 65 in their data. So that looks promising. And then there is this study in the New England Journal that looked at immune responses in transplant recipients who received the 3rd dose of the Moderna vaccine. And you can see that there was a higher titer of anti-receptor binding antibodies and of neutralization antibodies. So it looks good, but we need clinical data. No talk in COVID would be complete without some mention of vaccine safety. There simply isn't time to go into details. This is an awesome website the CDC has. It's basically a one page. It summarizes the status of the different concerns, such as anaphylaxis, thrombosis with thrombocytopenia, myocarditis, Guillain-Barré, et cetera. The good news is that probably by now, 360 million doses of these vaccines have been given to people between December and August. And out of those, only about 0.0019% have died that have been reported. And I would venture to say that the majority of those deaths had nothing to do with the vaccine. Some of those people may have died from COVID, that they were incubated. Some people, many were elderly and were going to die anyways. You know, the mortality from this has got to be exceptionally low if it happens at all. So again, feel free to go to this website and look at the latest data. Variants of concern. All right, well, in the interest of time, we've pretty much shifted from alpha to delta. Now, delta has these unique mutations that make it probably twice as contagious and also reduce its the efficacy of the older monoclonals, and or at least the Lilly monoclonal, and the vaccines. And then Delta Plus is the one that people are worried about. It has this additional K417N, which could render it, you know, either more contagious and or more able to evade the immune system. So you've heard recently that the number of people that one sick person will infect with the Delta strain is about 7. This is the R0. Okay, the original strain, the R0 was about 3. It's now about 7 or 8. It's not quite as bad as chickenpox, contrary to initial reports. Chickenpox is an R0 of 10, mumps is 12, and measles is 18. So we still have a ways to go, but this is bad enough. This shows you the CDC's basically their projection through August 28th in terms of the lineages of the viruses in the US. And you can see that the orange here is the Delta variant, and it's pretty much almost 100% now. I think here it's 99%. So it's basically taken over from the B117 or the Alpha, which was the previous variant that we were concerned about. Now this has outcompeted this one, and it's replaced pretty much everything else. This is a nice website, outbreak.info, which shows that fortunately, the Delta Plus is very rare worldwide, less than 0.5% cumulative prevalence. If you look in the US, this is the graph. I'm sure the data is behind, but you can see that it looks like in August, the prevalence is going down as the main Delta virus increases, and it's well below probably below 0.6%. So we'll have to keep an eye on that. The other one that people are fearful of is this Lambda variant that's lurking out there. This is a nice diagram that shows you the virus and where the mutations are and the potential impact of these mutations. So this is the N-terminal domain and the receptor binding domain of the virus. Red arrows relate to infectivity, and blue relate to neutralizing antibody resistance. So that would be resistance to the monoclonals and or the vaccines. And what you see here is, for instance, this 253N seems to increase resistance to neutralization by antibodies, but has no effect on infectivity, whereas the 452Q increases infectivity and resistance to neutralizing antibodies. And there's a number of others around. And so it's this combination that's kind of worrisome. And you can see that this spring, there's been massive spread in South American countries. The good news is, once again, the Delta variant is outcompeting everybody else. So in Peru, 65% of the cumulative isolates have been the Lambda variant, but in the rest of the world, it's less than 0.5%. In the US here, again, it's very low. It's less than, way below 0.1%. So I'm hoping, although you see there's a wide confidence interval here because of small numbers of reports, I'm hoping this means that it will not, but only time will tell. Who wins the Darwinian battle here. But hopefully, as bad as Delta is, I hope that that's the one that stays so we can deal with it and get this under control. So finally, I'm going to end on a sobering note. This is an interesting viewpoint from a few weeks ago in JAMA by 3 people at Emory and Brown. They talk about the endgame scenarios for COVID-19. So eradication refers to permanent reduction to 0 cases, the example of which would be what we've done with smallpox. Elimination is reduction of cases to 0 in certain geographic areas, but with ongoing control measures. So the example of that would be polio. Polio is gone from most of the world, but there are a few spotty outbreaks. Cohabitation is living with the virus, but managing pretty well. And conflagration means ongoing surges, tremendous social, financial disruption. Unfortunately, the authors, and I would tend to agree with them, are saying that over the next couple of years, eradication, elimination are highly unlikely. We're probably looking at cohabitation or conflagration, and we're really hoping that it's cohabitation. So cohabitation would be more or less like the flu, where you get vaccinated every year. If you get sick, you can get oseltamivir. Most people do pretty well. Some people do die, but you basically are used to it. You get used to it. You cope. Conflagration would be, that would be unpleasant. That would be, something that could interfere with travel and, would have probably higher mortality and would be spotty around different places. And that would be very unfortunate. So I think we're all hoping that cohabitation is the way we go. These are my favorite websites. I think the NIH is probably the most up-to-date. IDSA is very good. I also like the CIDRAP website. It's a great clearinghouse for literature. If you want to know what the latest studies are, and they often do an analysis that's great. And then the IDSA Real-Time Learning Network is a treasure trove of information. They basically do literature reviews for each intervention. So if you want to know what the data is behind, say, remdesivir, go there and they summarize it. And then, of course, the New York Times and the COVID vaccine tracker. This is the updated medical letter chart or table. It's pretty good. It's kind of busy, but it did cram a lot of data into that. So for those who want it, this is how you get it. And these are some great slides from the August 13th Council on Immunization Practices that meets every couple of weeks to review the latest data to make recommendations to the CDC on immunizations. And with that, I will stop and see if there's any time for questions. Looks like we do have some time.
We do have some time. Thank you very much, David. This excellent overview was very helpful. There is one question. Where can we get a copy of Dr. Mushatt's slides? This includes excellent responses to vaccine hesitant patients. I think actually they are actually recorded and posted in the Tulane website. Is there any additional comment?
Well, I'd be more than delighted to take emails from people. Just email me, dmushatt, mushatt @tulane.edu. Happy to send them to you directly. Please feel free to share them, copy them, whatever you need them for.
Dr. Cummings is asking any idea about the duration of efficacy of the third COVID vaccine dose?
No, although not really. I did see a report. There's actually an MMWR, I think a couple of days ago, there is a report where it's from, I think it's from another country, where they looked at antibodies in people who had SARS-CoV-1 up to 17 years ago. And interestingly, the majority still had antibodies. And when they gave them a SARS-CoV-2 vaccine, I think, they seem to have very broad responses across clades, which bodes well, which means that, by boosting, we may be able to get better protection against not just the current mutant, but other mutants and other similar coronaviruses. But right now, no, there's antibody response data because the third doses have not been used for very long. They have been used, but there's not much clinical data. So the jury's out, but it does look promising.
Kelly is asking thoughts on counseling patients about booster dose for those fully vaccinated patients who have subsequently developed a breakthrough case.
Well, I think in general, we vaccinate, regardless of whether somebody's had primary infection or a breakthrough. There are studies that show that if you've had COVID and then you get even a single dose, but ideally 2 doses, you get much higher antibody levels. So you probably get even better immunity. But just be careful. Remember, we don't yet know how well antibodies correlate with protection because there may be, the antibodies we're looking at, neutralization antibodies, anti-spike, may not be the whole story. There are T cells and B cells, memory cells in the background that probably play a role. That's much harder to get a handle on, so that it's conceivable that antibodies will go down, but people will still have pretty good anamnestic responses, memory responses. So it's a very murky area, but bottom line is, if you've had a breakthrough, you can still get the booster. The general rule of thumb is if you have COVID, you should not get a dose of the vaccine, either your first one or your second one, or the booster, until you've recovered. So, and you're basically out of isolation. So that's basically 10 days. I think a lot of docs feel more comfortable maybe waiting about 4 to 6 weeks before they give that dose. And then of course the CDC says that you can wait 90 days if you want to, because there's probably pretty good protection 90 days out.
Dr. Schamberger is asking, are monoclonal antibody treatments recommended regardless of the vaccination status if the patient meets the risk factor criteria?
Absolutely. That was my first question a couple months ago when I first had an opportunity to use it. And I did a literature search because it was a breakthrough. And I thought, well, maybe it's not indicated. And there were already reports from New York of people who had breakthroughs who got it and were, clinically improved within 24 hours. And so, yes, now they are recommended regardless of vaccination status. There's not a lot of data, of course, on how well they work in that population, but they do appear to be safe and effective.
The next one is from Dr. Hua Lu. Great talk. Any new clinical trials for small molecule antiviral proteases going on right now?
I don't know. I mean, there's so many things out there. I do know that we're part of the Active2 adaptive platform. We've been trying to get it off the ground now for almost a year, but due to regulatory issues and closing of the various arms, we haven't been able to get anybody on the studies. And one of the drugs was the Camastat, the oral protease inhibitor. My understanding is it failed in phase 2 and is not moving to phase 3. There probably are, I'm just not aware of that. Molnupiravir, I think, is the furthest along. That's the ribonucleoside analog. Merck is saying that it may be available in early 2022, but of course the clinical data is key, because not everything that lowers viral load necessarily has improves outcomes.
Dr. Palomino is asking, could the difference in results from the anticoagulation trials be due to timing?
Yes, I think that's one possibility. Again, read that editorial. It's really well done. And also read the heparin resistance editorial, because one theory is that people with COVID may have more clotting factors or substances in their physiologic milieu that actually make them more resistant to heparin or low molecular heparin, and that probably is more of a problem in more advanced disease. So I don't think anybody knows. And again, I'm not a clotting expert. I really always defer to Dr. Lessinger, and frankly, the hospitals probably know more about anticoagulation than I do. But yeah, it's a very frustrating area, because I think we'd like to believe that prophylaxis or therapeutic treatment would help, but we still are seeing people that break through and get PEs on heparin, and so maybe there is this resistance phenomenon.
Denage Bostic is asking, is there potential for the Lambda variant to become more prevalent than the Delta variant worldwide?
I wish I knew. You can ask Bob Gary, who's a world-renowned virologist at Tulane. I suspect he'll say, hope not. My understanding is we still don't really have really effective computer models of viral evolution in terms of predicting. I mean, there are some models, I understand, but they're just not at the point yet where they can still really predict what mutations are going to occur, and if certain mutations occur, what the effect is going to be necessarily on the virus vis-a-vis the host. So we really can't predict. But again, my hope is that the current mutant will outcompete those, but it's also possible. One concern that the WHO has about the US embarking on boosters is it's going to use up vaccine that could be used in other countries. And I get that. That's an issue. So as Vivek Murthy says, it's not either or, it's both. We should be giving vaccines to our population and shipping hundreds of millions of doses overseas. So it is a little bit of a dilemma, but it is possible theoretically, I suppose, that if we vaccine the heck out of this virus, we could create selection pressure for Lambda. But I think that, the jury is out. We just need to do as much sequencing as we can to keep an eye on this.
I think we're coming to the end of our time, but we can take one more questions. Cesar Aguilar is asking any comment about mixing different types of vaccines as part of the booster strategy.
Yes, I haven't seen anything recent in the US on this, but I do know that there's data overseas where they gave second doses of mRNA vaccines after the single J&J dose, and they saw good antibody responses. This is essentially heterologous vaccination, which is a long-standing principle in vaccination where you give doses that either use different parts, say, of the adenoviral of different adenoviruses, or you give completely different platforms, and it may actually be that that's better. I don't know. I don't think there's been a heck of a lot of progress in that here, but there should be more data fairly soon from overseas because we haven't used much J&J here. Pfizer is the #1 vaccine, far and ahead of J&J, and Moderna second. The number of people getting J&J is pretty small, but the AstraZeneca vaccine's been used a lot in UK, and there should be more data. But yes, that should be doable. It's just not officially authorized in the US.
I'm sorry we don't have time left to be able to answer additional questions. A repeated question was about the recording. It will be made available. Give it some time. I don't exactly know, maybe in about a week or so. All the grand rounds are posted on the Tulane Medical School website, so you can find them there. I want to thank nearly 130 participants and our wonderful Dr. Mushatt for this lively discussion and update. Thank you all, and have a good afternoon, everyone.
Thank you, everybody. Thank you for your time.